But there's a very, here. So the cable, if you see, this is a cell which is confined to sit in a circular shape. Okay, so these are actin, which is getting organized into this cable-like structure. Now, what happens between two cells is these actins interact with each other so that you form a supracellular cable-like structure. So that typically could look like this. This is at a single cell.
What's the role of this curvature of the boundary?
So the leader typically, people have been trying to look at curvature-related proteins because the polarization will induce the curvature-induced proteins to sit at the front edge of the cells. So there is a correlation that polarizations in the high convex regions will create the leaders. But how does it come to pass that the polarizations occur there is not very well understood.
When the leaders emerge, I was wondering if you see some local tidal flow when the leaders are emerging.
Yeah, that's a good question. So actin is a chiral molecule. So in fact, when we put our cells inside this confined space, what you see is that there is a lot of chirality that you see a flow in the structure. So in the presence of the leader, we don't see as much because the leader has a lot of lamellipodial regions where you have very small, short fibers of actin which are pushing forward. So you won't see as much of the chirality that you see in the interior cells.
On the time scale of your experiments, the cells are dividing.
Yes.
Are they also dividing?
It's a good question. We don't see cell death, but to be honest, we haven't measured. We haven't measured. We've only kept track of it. It's an excellent question. Is with regards to those defect structures, do you see any deaths? But no. But based on what we can measure with the aerial strains, we have pretty much doubled our monolayer. So it suggests that if any, there are a really small number of cell deaths.
Another question is that when you compare the motion of a single cell versus the collective motion that you see, which one is faster or are they the same?
So also a good question. Most of our previous work, we have created race tracks to make cells run fast on microcapillary. And so with that, we can see that single cells move at these cells move at almost 7 micron an hour. These cells are much slower moving as compared to individual cells because I think of the baggage that we have to carry.
And with the baggage, you mean like
the additional cells because they can't come off and crawl alone. So, they're slightly slow, but and that also reinforces the adhesion between the two cells, which becomes relevant.
Thank you. Thanks, Namrata. We should now move on to the next talk. Our next speaker is Pranay Mandal from Max Planck Institute of Molecular Cell Biology and Genetics. And Pranay will talk about protecting DNA against mechanical stress.
আমি শেয়ারটা বন্ধ করি। Sure. এটা লেজারটার সাথে ইউজ করে আর ওখান থেকে চেঞ্জ করো তখন এখান থেকে ওনাকে ফোনটা রেখে
So, hi everyone. Thanks to the organizing committee to give the opportunity to present the work in front of you. Today I'll be presenting my recent postdoctoral work, which is about finding how cells protect DNA within it. And my work is mainly in vitro reconstitution, which I have done in optical tweezer, and these results are backed by cell biological experiment and some theory. But due to time restriction, I'm afraid that I can't share those results. But mainly, I'll be sharing the in vitro result. So, as you all know, that it's very important to protect DNA because from the DNA, information flows to create, synthesize proteins and other things. So, but in nature, there exist different kinds of stresses that leads to DNA damage and that leads to cancer and other problems. However, I'm going to talk about the mechanical stress that is being generated, and that leads to mainly three kinds of DNA damage, mainly the double strand break, where both the strands of the DNA break, the single strand break, or the melting, where the hydrogen bonds between the nucleotides break. Now, before I move forward, I want to make sure that the mechanical stress that I'm talking about is very much practical. In fact, in the real life scenario, where this is one example where the chromatin being pulled by the microtubule during the mitosis process, and this this pulling can generate a force of 100 piconewton. And also, like this is a famous example, like that there are a lot of heart diseases that are caused by the DNA damage that are caused because of the mechanical pressure from the heart cells. And also there are some external reasons which can also cause DNA damage. Here you can see the cell migrating through some constricted space. And here, when the cell passes through this restriction region, it causes DNA damage, which are shown by the DNA marker here in the red. So the mechanical stress indeed causes DNA damage, and the question is how the cell protects it. Well, although the DNA damage can lead to cancer, we are lucky enough that the cell has some DNA repair path which can keep the DNA intact. However, as we say that like prevention is better than cure, is there any way we can protect? We can stop this flow, or in a way, is there any way that we can buffer the mechanical stress that can protect the DNA? So indeed, there are a lot of mechanisms, but one of the mechanism is the protecting the DNA by the nuclear envelope. The nuclear envelope is a membrane layer which protects the DNA from mechanical stress. However, this nuclear envelope goes away during the mitosis. So during the cell division, this nuclear envelope goes away, and the DNA is exposed to the external stress again. Also, there can be events where there can be nuclear envelope rupture, and that in that case also DNA is exposed. So the question is, what protects the DNA in the absence of nuclear envelope? So to understand this question, let's see what happens when the nuclear envelope goes away during the mitosis in this case. So here, what you see in the magenta color is the chromatin being pulled by the microtubule during the cell division, and you can see that the DNA, the chromatin, is being coated by a cyan-colored protein, which we call BAF, the barrier to onto factor, and it seems that it kinds of coats the DNA and provides some protection to it. There are also reports where, in case of nuclear envelope rupture, the BAF comes and coats the nuclear envelope. So it seems that it has some protective mechanism. Before I move forward, let's explain. Let's see what's the structure of the BAF. The BAF forms a dimer, and it can bind to DNA, and it has some interesting feature that it can bind. The BAF can bind to another protein which has LEM domain, and LEM2 is another. LEM2 is one such protein which can bind to BAF, and BAF binds to chromatin. So it's a cascading structure where it can bind to chromatin and also to the nuclear envelope. And it shows different types of interesting features, like it has intensively disordered region where which can show protein-protein interaction, and it can phase separate. Now, why I'm telling is there are reports where if you silence BAF, what we see here is the single nucleus breaks apart and there is a formation of multi-nucleus. So the function of the nucleus goes away in the absence of BAF. Similarly, when you silence LEM2, there are reports that it develops DNA damage, which are shown here with the cyan color, and that happens in the absence of LEM2. And BAF and LEM2 binds to each other. So it seems that these two protein complex provide some worsening effect, surface tension, and some protective mechanism. So our hypothesis is this: like the BAF and LEM2 protects DNA from the mechanical damage. Now, in order to prove that, we will use optical tweezer. In the optical tweezer, what we can do is we can catch a single piece of DNA with bioconstructor with functional relation, and here you can see like we can also stretch the DNA by optical manipulation of the bead. And here the video is like is a single DNA getting stretched, which follows the worm-like chain model. It's just like a polymer, and the DNA is being coated with a dye which only binds to the double-stranded DNA. So we can also tag protein with the fluorescent dye, where we can also study the protein binding dynamics of the protein to the DNA. So let me come to the result. So what we see is when we add BAF and LEM2, the LEM2 is labeled with the red color here. And what we see when the DNA is stretched to some initial force of 40 piconewton, we see that there is an increase in force of something around 7 piconewton, and that 7 piconewton is against 40 piconewton. For the biologists, like I want to tell that like this force is a humongous force, and I will give you some numbers in the upcoming slides. And this increase in the force, or there is some energy released, that is without any ATP or GTP, and this is just plain thermal and normal binding in the equilibrium condition. So the question that, and also what we have seen is, if we take different lengths of DNA, the increase in force per base pair remains the same. And that is shown here in this curve, and that which means that something inherent property of the DNA is getting changed because of the binding. To understand this process, what we have done is like we have done a constant force experiment where we keep the force constant when we allow the protein to bind. So when the protein binds, the force tries to increase in the system, and we have a feedback mechanism in the optical tweezer, which gives a slack to the system, and that's how the force is getting is remaining constant. But what we are measuring is the compaction in the length. And when we plot the compaction as a function of the initial force, what we see as a linear increase. And what it tells that more the polymer is stretched initially, more is the compaction, and it varies linearly, and it also varies with the concentration. So we try to understand that, but before like this is the number that I was talking about. Like the compaction that we see here is about 250 nanometer, and the force is about 40 piconewton. So if you do the number, that turns out to be this 2500 kT, and that is equivalent to burning of something around 125 ATP molecules, and that energy is being generated just from the binding energy of this protein. So one way to understand this compaction is like the stiffness of the DNA is getting changed, it's getting increased. So in the polymer, this slope of the force distance curve is basically will give you some rough assumption of the stretch modulus of the polymer. And what we are seeing is this binding is actually making the DNA more stiffer, and it changes from like 1300 piconewton to 1900 piconewton. But what's the molecular mechanism for this? So in order to understand this, what we have modeled this system where the DNA is modeled as a series of springs with some spring constant K1, and the protein complex, which is like 2 BAF with some with LEM2, because the LEM2 also interact with each other. So there are more than 2 LEM2 here, but the BAF is interacting with each other via LEM2. And that is we are modeling with some other spin constant, some K2. And this is the whole system. Like this is the DNA with some series of springs with K1 and the protein complex which are also springs in parallel, and that's the thing. And we can calculate the the energy of the microstates and with some SFP constant. And from there, we can calculate the partition function of the system with some standard approximation. What we try to do is we solve for the equation which minimizes, we solve for the equation of the end-to-end distance of the DNA, which minimizes the free energy. And when we do that, we indeed get that the slope when the force so we let's concentrate on this region. What it tells us that when the force is increasing the end-to-end distance, the stiffness of the polymer also increases. But what it also tells us that if we keep on increasing the force, the stiffness should again decrease. But this is beyond the experimental limitation, because the DNA breaks before this force reaches, but it captures the increase in the stiffness of the DNA at some lower force below 60-65 piconewton. The formula also this formulation also captures the increase in the compaction with the initial force, which goes as linearly with F. Now, it's now I've said that the DNA gets stiffer with the binding of this protein complex. But what about melting? Now, like the DNA gets melted at around 65 piconewton, and that is being shown in this video, where we kept on stretching the DNA, and at the 65 piconewton, that is the universal force at which DNA melts, we get a single-stranded DNA, and that is shown here in the dark spot, because the dye that we are using only binds to a double-stranded DNA, and what we are observing is the melting at 65 piconewton. So that's the signature of the DNA melting, and that we should remember. And similarly, we do the same stretching curve in presence or absence of the protein complex. But for the time being, let's concentrate on this part. So this is the melting signature where it gets flat and where the DNA is melted. And you can see here that the only DNA melts at 65 piconewton, but when we add the protein complex, the force increases. And this is indeed the case with the BAF and LEM2 complex. The melting force is about 20 piconewton higher than the normal force. So that's the BAF-LEM2 also prevents the DNA melting. And the last one is like what happens to this scenario is like when we relax the DNA, it forms co-condensate with the DNA, and this is the signature of the phase transition, because no matter how much we relax the DNA, the force gets clamped to some particular value, and that's the signature of the first order phase transition. We have also characterized this, and we can calculate the free energy of the condensate, which is like the free energy, the volume, and the surface. And what you can see that the force indeed goes constant when the transition happens. And I will just keep this. So what we have, so this co-condensates can actually prevent the DNA damage by force circulation. And the idea is when the DNA is being pulled by the microtubule, the force actually goes through the DNA. But when you have this condensate forming by the BAF and LEM2, the force actually percolates through the condensates, and the DNA is no longer facing the higher force at which the microtubule is pulling. And we have done some control experiments where we have seen that the DNA damage indeed happens outside this condensate, and the DNA is protected inside the condensate. So we started with this scenario where the BAF-LEM2 can protect the DNA, and there are three mechanisms which makes it stiffer by binding. It opposes DNA melting, and it undergoes first order phase transition by forming condensate that can also protect the DNA. So I would like to thank I'm doing postdoc under Stefan Grill, and I'm collaborating with Alex and Frank, and these are my collaborators. And thank you.
Thank you very much. One quick question.
Yes, you see do you see a difference in your localization of this protein if the cells are under stress, any kind of mechanical loading, where you see heterochromatic condensations?
So what we have seen is we have silenced the protein in the cell, and we have compressed the cell. And what we have seen is indeed that there are a lot of DNA damage, and there are proteins which agglomerates at the plates for the DNA plates. So there is some local accumulation of the protein.
So the mechanism is to preserve the DNA, and so it's sort of
Keep on the nuclear
on the
envelope.
nuclear
Yeah,
membrane.
yeah.
Okay, thank you, Pranav. Let's move on to the next talk. Next speaker is Mridmoy Mukherjee from Northeastern University, and he will talk about the alternate research jagged enhances robustness of Mach signal devices.
Last. Hi, can you hear me properly? Yeah, okay. So my voice has been choked, so that's why to confirm. Maybe you have to bear with this different kind of voice. Okay, today I'm going to discuss about the cell-cell communication in a epithelial tissue, mainly through notch signaling pathway. How the spatiotemporal pattern emerges and what are the possible role of different ligand, especially the jagged one. So notch signaling pathway is a typically in other biological system, the pattern formation through the reaction diffusion system, it is little bit different. So this is kind of a juxtakin signaling instead of diffusive signal, diffusive chemical. Here it only works when two cells in contact with each other. So these govern the emergence of spatiotemporal patterning in different contexts of development, wound healing, or any kind of collective cell migration, or even the cancer metastasis. So today's I will only focus on the spatiotemporal pattern in the context of development, mainly in epithelial tissue. So here the ligand delta generally differentiate the cells in spatially. So there will form some high delta cells surrounded by low delta cells. So I'll discuss why the high delta cells are specialized. Typically, the mechanism is when two cells are contacting each other, the different kind of ligand, here delta and jagged, bind to the notch receptor, and this notch intercellular domain, NICD, cleaved, and it transferred to the nucleus, and there is some transcriptional regulation which either increase or decrease different level of receptor and ligand. So we can differentiate. There are many kind of notch receptor or different kind of ligand according to the mammals and other animal also. But we can differentiate in two categories. There are delta kind of ligand, which form the lateral inhibition kind of pattern. Which so if there is two neighboring cells, one is a high delta, it always try to inhibit the neighboring cells. So we'll get a divergent cell fate starting from a homogeneous condition with some fluctuations. We'll end up in this kind of state. And on the other hand, there is another kind of ligands, namely the jagged, which go through the lateral induction pattern. So if there is a high delta level in somewhere, so it's spread along all over the cells. So this is a typical example of early development of chicken inner ear. So here you see the white cells are high delta cells surrounded by the low delta cells, and you will see it is almost perfect. So it is almost perfect in pattern. Here high delta cells are called sender cells, and we will mainly for the talk we will stick to this notation, the sender cells and the surrounding cells are receiver cells. It is called different name depending on the context. So our thing is what will happen in presence of both lateral inhibition and induction. Is there any synergy, or these two effects always oppose each other? And what are the possible role of jagged in the order and robustness of this lateral induction pattern? Because in reality, we'll see very less error in this system. And I will see, we'll see from this model, if we simulate, then there is always some kind of defect in the patterns. So we use the simplified model of notch, delta, jagged, and NICD level. So there is some kind of production rate we implement it as a hill function, and obviously this gamma is the natural degradation rate, and it interact with the neighboring cells through trans interaction. This KT is the trans interaction parameter, and instead of trans interaction, there is some kind of cis inhibition also. So the same inside the same cell, notch and delta can bind. This does not produce any signal, degrade the level of the signal. So here we take a epithelial layer of hexagonal symmetry. In most of the cases, epithelial tissue is close to the hexagonal symmetry. It is not exactly hexagonal, but for the simplicity, we have taken the hexagonal symmetry. So now the strategy is, first we try to solve the equation, the linear stability analysis to get the phase space, and do the simulation accordingly for all the cells. Now, because of this hexagonal symmetry, instead of solving if there are n cells, instead of solving say 4n square equation, we need to only solve the 12 equation, 4 into with these 3 unit cells, because this ABC unit cell expand the whole radius. We'll focus on these hexagonal ordered patterns. We'll vary the production rate of N, D, and J, that means lambda L, lambda D, and lambda J, and check the order and robustness in the pattern. And for all the other parameters, we use this the range of parameter from this experimental paper with this some synthetic, not single. Typically, in a hexagonal lattice, we can get two kind of pattern. So one is called we define it a hexagon. That means high delta cell is surrounded by the high delta cell rate, surrounded by the low delta cells, and opposite to that, it can happen that low delta cell surrounded by the high delta cells. We define this uniform as U, where the difference is 0, hexagon, where the delta D is greater than 0, and N and D in a cell is always opposite. So if delta D greater than 0, that means delta L less than 0. Opposite, similar to the anti-hexagon. And we define this delta as difference between the red minus blue cells. And of course, these red cells, we call it sender, and blue cells the receiver, because red cell send the signal effectively. Now, if we calculate this bifurcation point, so as at a fixed value of lambda J, very small value, say for the first, and now as we increase lambda D, the production rate of delta, the uniform stable phase go through a transcritical bifurcation to a unstable hexagon and unstable anti-hexagon, but eventually the unstable anti-hexagon go through a stable anti-hexagon to a saddle node bifurcation. And this is imperfect bifurcation. The important point is that for a range of lambda D value, we can get a bistable phase. We'll discuss later what is the implication of this bistable phase. Of course, we can always get a perfect bifurcation for a particular value of lambda L. For a particular value of lambda L, we can find a particular value of lambda L so that we can get a perfect bifurcation point. Similarly, we can find the opposite case when this lambda is greater than this pitchfork, greater than this lambda pitchfork bifurcation point. We'll find just the opposite of this thing. So now we can calculate the phase space through this calculations. Oh, by the way, one quick reminder is that in nature, in biological system, we generally not, people can't find this anti-hexagon kind of solution. It is always hexagon kind of solution. So now, as a lower value of lambda j, when it is almost negligible, we can, this is the phase space according to lambda n and lambda d. So we can get a uniform phase, left side of this black region, and this is hexagon phase, and this is inside this blue line, it is anti-hexagon phase, and we all sorts of couple phase, bistable phases. As we increase this lambda j, two things to observe here. We find this region, we find a bistable region, as we discussed previously, and this region for anti-hexagon is decreased. So we can argue that there is a low likelihood of anti-hexagon phase in biological with insufficient parameter control, because this region becoming narrower and narrow. We don't know the exact reason for that, why we can't find the anti-hexagon in nature, but this can be possible reason, because to control the parameter in a narrow space will be difficult for noisy biological system. And now we will focus on this bistable region. What is the special about bistable region? How can this ordered pattern generated in a noisy biological system? As we discussed, so now if we simulate here, there are 2,500 cells, 50 cos 50. So now, if we solve the four equation of notch, delta, jagged, and NICD for all whole cells, so you will see this the pattern nucleates from different places of the lattice, and they grow, and there is boundary mismatch. You will always find this kind of disorder. So this obviously depends on the system size. For the small system size, the probability of getting another nucleation center is very low. So we'll always get a perfect ordered pattern. And for the very large system, we'll always get a disordered pattern. And so we can simply calculate this number of sender state, that means red cells by blue cells. And for a perfect ordered pattern, it should be half, because it is just, you can think of ABC unit cells. So 1A is the sender cells and BC is equal. So it is 1/3 by 2/3. It should be half for a perfect for a perfectly ordered pattern. As we see, it decreases, but it can always improve with increasing the lambda j value. We'll discuss later why. Now the implication of this bistable region. Now, if we choose a parameter space from this bistable region where the uniform and hexagonal phase both stable, and we start from a seed. So here the fluctuation, other than this seed, does not grow. So the seed only grow, and it spread as a perfect pattern. So we can say that if we somehow choose, if the biological system can somehow choose some parameter space from this bistable region, we can get a perfectly ordered pattern. And the implication of this is, as we increase lambda j, the width of the bistable region increases. So without jagged, this bistable region is very narrow, as you see. So enhanced region of bistability can be a useful strategy to obtain accurate pattern in noise biology system, because you have enough choice to choose the parameter inside a large bistable region. Other than that, there is a very interesting property of this dose-dependent role of jagged. That means the production rate of lambda j. As you can expect, because jagged produce always the homogeneous pattern, and delta produce the alternate pattern. But at low dose, if we see this, if we calculate the difference between red cell and blue cells, you see this. Initially, it actually increases. So that means the pattern become more refined. So this, the reason for this, the competition between delta and jagged over binding the notch increase the delta in center cell compared to the receiver cells. So actually, the jagged act in synergy with delta to refine the pattern, especially at low dose. After some time, it becomes opposite. So you can think of this picture. So initially, if you have a two cells, the low delta cells and high delta cells with this gap delta D, as we increase lambda j, both increases, but the increment of the high delta cell is higher, so that's effectively delta D increases. But when we enter in this bistable region, another hybrid state emerges, and eventually, when lambda j is very high, we will find one homogeneous ring. So the summary is we show that jagged, which has the opposite property of the lateral index, lateral inhibition, can act as a synergy at low dose to get the perfectly ordered pattern, and there is some role of this bistable region in to get the defective. Obviously, there are many other biological strategies, especially the mechanochemical coupling and other thing also to get the perfectly ordered pattern in the developmental cell. But this jagged kind of strategy can also be a mechanism. Thank you. You can find the paper here.
Any questions? Time for a couple of requests.
If the cells were square, it would be like anti-thermomagnetic, I think. So now this hexagonal, and then there are three types, and then the ABC alternate. Do you think of any statistical physics model like this?
Sorry, I don't understand.
I mean, so if the cells were square, I mean, there will be AB, AB, AB. It will look like anti-thermomagnetic, I think.
Yeah.
With this particular thing, can you think of any statistical physics model which will go to such kind of state, lattice model?
No. What do you want to explain that? But one thing is, in square lattice, the thing is there will be no external pattern, and also the biological system is not square. You are right that there can be some model, but I don't know what it is.
Also, you have so much more than irreversible interactions. It's much more complicated.
No, but in this kind of signaling, only the nearest neighbor kind of interaction work. But sometimes if there is laminopodia or philopodia, sometimes an extent, so they can get up to 2-3 cells. So you can get some kind of sparse pattern, but not more than.
Okay, so let's thank all the speakers of this session. And now we will go down to the lawn for the group photography.
Yeah, that's it's okay. So it's just it's okay. And the last one, this one, yeah, this one. Copy or cut. You just check, sir. Sure. Is there any video? No. No video. No. Yeah, it's okay. Just using this one. It's okay. There is some problem? No, 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 no problem. Okay. Okay, sir? Yeah, yeah. Second. Wait for the photo. Sure. You go? Yeah. Thank you very much.
So they have a sticky.
মানে এখানে
Yes, 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 yeah, yeah, so it's not such, yeah, yeah. So these are not
Yeah, download it.
Download. ওখানে হ্যালো বলো
ট্রান্সফার মানে এখানে মানে ডাব্লিউ ইন্টিগ্রেটস এটা হচ্ছে ইচ ডাব্লিউ আর একটা ইচ ল্যামডা দিয়েছি আমরা ল্যামডা এটা কোনো ইন্টারেস্টিং এডিশন করেছি মানে এই ওয়্যারের জন্য আর এই হ্যামিল্টনিয়ান করেছি আর এই ল্যাবটা ডায়াগোনাল ট্যাবলেট আছে এটা তো দেখতে পাচ্ছি যে দিস কমপ্লিটলি অফ ডায়াগোনাল আমি কি একটা পেজ থেকে আরেকটা নিজেকে নিজে কানেক্ট করতে পারি এইটা কিন্তু ডায়াগোনাল ট্যাবলেট কারণ কি এটা যেহেতু এটার উপর আবার রিলেশন কি প্লাস দ্য সেম স্টেট সামেশন অফ ট্যাবলেটস
Okay, here we go.
এবার ল্যাবটা যখন লার্জ পজিটিভ তখন ট্রান্সফারেন্স যে ওরিয়েন্টেশন সেগুলো ম্যাক্সিমাইজ করা যায় কারণ এটা মাইনাস ল্যামডা হ্যাঁ এটা ব্র্যাকেট আছে ল্যামডা যদি লার্জ পজিটিভ তাহলে সেটা কিন্তু মাইনাস আছে ও ট্রান্সফারেন্স কনফিউশন গুলোকে ম্যাক্সিমাইজ করা যায় আর ল্যামডা যদি লার্জ পজিটিভ হয় তাহলে ওই লং ডিগ্রি ম্যাক্সিমাইজ করা যায় তাহলে লার্জ পজিটিভে ল্যামডা লার্জ নেগেটিভ যখন হবে তখন এই স্ট্রাকচারটা ইজ দ্য অনলি গ্রাউন্ড স্টার এখানে ম্যাক্সিমাম ইয়ার দিস ইজ এ ইউনিক গ্রাউন্ড স্টার আর এটা হচ্ছে গ্যাপ এই ইয়ে লাইনে এই ল্যামডা লার্জ নেগেটিভে যাস লার্জ নেগেটিভ দিস ইজ এ গ্যাপ গ্রাউন্ড মানে ফার্স্ট সাইড থেকে কোনো ক্রস করে নেই এটা গ্যাপ গ্রাউন্ড
Sir, your good name, sir? Your good name, sir? Your good name, sir?
Prashant, Gade. GD entrance.
ম্যাক্সিমাম নাম্বার অফ ট্রান্সভার্সটা এইটাতে হতে পারে এরকম একটা স্ট্রাকচার এরকম একটা স্ট্রাকচার
Sir, just check.
Yeah.
Okay.
Second power, immediately will be. Fourth power, yeah. Yeah. Actually, you can do the exam question. And that's why. So. History about 0.2 or 0.3, depending. I give you no, it's definitely. Yes. So this is the one probably, right? For the factory, so there are important initial condition that okay. If start from a factory, they usually they get the remain traffic numbers. I don't know is that in that case, the expecting student is about 0.20. But if you start from a point and get the factor
configuration maximize
so if we are in front of but and you see the depend on in here case it's intermediate, so they start from a factor go to the boundary you expect it gets stopped.
but
I don't know which case. That's why it's more in any case that is it's not valuable.
এখান থেকে বলা যাচ্ছে যে
And the experimental verification that
পার্টিশন পয়েন্টটা আমরা দেখছি মাঝখানে একটা ল্যাপটা সি পয়েন্ট আছে ল্যাপটা সি পয়েন্টে কি হচ্ছে মানে ফেজটা মানে কিরকম সেটার জন্য আমরা স্কেলিং অ্যানালিসিস করছি she's scaling up this system that even a fourfold each point day. Ground state, executive, first state, second state.
Oh, compass. Compass is also very.
Cut kursi. Fourfold degeneracy. Like each fourfold degeneracy, you know, and minimal value.
So
He's
these are.
coming up to 1.55 something.
I think your case, satisfy our criteria.
The controllers are half, I think, critical rate. I think critical. Controllers are
But my experience is that one
actually, we actually
I mean,
from the calculation of the C conformance calculation to feel scaling analysis. It falls under the Ashkin but then it comes একটা Z2 হচ্ছে তোর একটা asymmetry আছে সেই asymmetry টা হচ্ছে এর আগে একটা symmetry আছে reflection দিস তাহলে একটা symmetry সেই asymmetry টা দেখো প্লাস একটা Z2 হচ্ছে emergent সেটা কি সেটা বোঝা যায়নি an emergence symmetry টা বোঝা তো পরেই যেটা হচ্ছে I think
yeah, because in such a case, what so you decide to do that you can try you group quantity not against time but against the mean time. So when you take average, you don't take average of data at the same time, but at the same height, same mean height. Then you can kind of get the same. Of course, because mathematically speaking,
no, but I know. So another quantity.
এখানে সিগমা আর মাঝখানে যেটা টাও
yeah, so actually, right. I mean, I thought you
Another experiments, I just want to see. Hello? To say. Yeah, I'll just. Hello? Just one second.
একটা লাইন ফিজিক্যাল সেফিশিয়েন্টলি চেঞ্জ করছে নরমালাইজেশন কনস্ট্যান্টলি মানে কনস্ট্যান্ট হিসেবে সিগমা সিগমা after surrounded by other things. And each of them surrounded by other. মানে একটা A dot surrounded by B, B is surrounded by A.
You just say, I just take such this event
maybe I know. There is such a difference. Yeah. The software tablet. The previous tablet. And then yeah, it's for the HTML file. I just check there are such periods. Period ratio administration. I think. Just as both are kind of So, right, energy, gentle. So, then the question is really imagine that different elements.
Welcome to the last session of the day. So now we have a talk from Yoon Chung from the University Korea, and he will talk on IDMRG for Hubbard model and IP model.
Good afternoon. Thank you for coming here. My name is Yoon Chung, and I work at Korea University, Korea. Title of my talk is Density Matrix Derivation Group for the one-dimensional Hubbard model and the island. As we all know, this model is completely solvable. So here, the main issue is that the model. Here, the main point is to provide usefully the power of numerical method, DML. So here, let's focus on the numerical. Okay, this is outline. First, I introduce numerical method, emphasizing on matrix product operation, and then I apply this method to one-dimensional Hubbard model, and I will also apply my method to island. So here, this DMRD can be applicable to the quantum case and classical case simultaneously. So that's the point, and I will conclude. So let me start by presenting some simple changes from sum to product. So Leibniz found that this formula, so pi over 2 is written 1, 3, 5, 7, 9, 11, something like that. And there is another formula called Wallis product. So pi over 2 is written as 1, 3, 3, 5, 5, 7. So please notice this addition here and multiplication there. So the same thing can be written as sum and product. Okay, then let's see the another example. 1D quantum eigenvalue. As usual, the Hamiltonian is written as this form. Sigma is the Pauli metric. This is a very familiar form, and this 1D quantum eigenmodel can be written in this way. Here, the site is only 4, but we can increase arbitrarily any site. So please notice this, the main matrix operator is 3 by 3 here, there, and there. And if we calculate this matrix product, then it will become 1 by 1, eventually 1 by 1 matrix. So this one exactly same as.
Excuse me. Oh, what is sigma 4? What is sigma 4?
This one.
No, sigma 4.
sigma 4. Here the site is 4, but you can increase arbitrarily many. Right? Here, so here 2, 3, 4, 5, 6, and this is the last site. Yeah. So it's I mean just let's consider a 4-site model. Yeah, but we can increase this site as many as you want. Okay, so another example is the Heisenberg. So Heisenberg model is just S star X. Everybody knows this model, and we can present this Harvard model as using this product. So, but big difference is the previous one is here I have to use 5 times 5 matrix operator here. So again, here is just a 4 time, but we can increase many. Then here there is a diagrammatic notation there, and the matrix has 2 indexes, like row and column. So the matrix has 2 indices. Then the matrix has 2 legs. In diagram, the matrix has 2 legs, and vector has 1 index. So vector has just 1 leg. This is the diagrammatic notation. So in general, this notation is called Penrose notation. So Penrose introduced this notation for tensor contraction. For example, there are 4 tensors, A, B, C, D, and this connected line means index contraction. So there is a connected line, 5 connected lines, 1, 2, 3, 4, 5. So summation is there are 5 indices summed out. Okay. And in this process, the order of contraction better for computational cost. So A, B, C, D, the multiplication A, B, C, D, or C, D, A, B is different in the computational cost. So, but it's easy to figure out which is the best. But yeah, and there are so many softwares. There are iTensor, Unit 10, Tensor Network Solver. There are so many software which is written by so many computer programs like C++, Python, and MATLAB. MATLAB is the endpoint, and Julia is the tensor operators. It's the Julia, and nowadays Julia is very popular and it's very convenient. But still, the iTensor written by C++ is very suitable for beginners. So this one written by Steve White and Student Meyer and Fishman. I recommend this iTensor. Okay, so as far as we know the matrix product operator, then it's natural to consider matrix product state. And in mathematics, it was proved that for given local gap Hamiltonian, the ground state satisfy area law. Area law means less entangled. The upper case is volume law. It's many heavily entangled, and area law means less entangled. So, in the simple tensor network states, I mean simply connected, then this state is less entangled. So it's natural to consider the ground state may be tensor network state, or tensor network state will be ground state. So let's take the ansatz for ground state with the matrix product state like this one. And we know the matrix product operator there. So previous, here is the matrix, but we have to change from operator to number. So this, for example, the S equal half, the state is up and down, and this state, this operator acts on this operator acts on this local pivot space. So eventually, our matrix product operator become 4-index tensor, like so there is a length 4, 1, 2, 3, 4, and MPS becomes 3-index tensor, like 1, 2, 3. And here yellow dot is the Schmidt coefficient. So we'll use this Schmidt coefficient when we calculate the antinormal entropy. Okay, so how to solve? Okay, so this is the main equation. I mean, this MPO, MPS, and there is a eigenvalue. So we have to solve eigenvalue problem. How to solve? That's the DMRG. DMRG is using this DMRG, we solve this eigenvalue problem. So first, we introduce environment tensor. So left environment tensor and right environment tensor. And this one is the MPO, the 2 MPO. And this is MP, sort of MPN. So we solved this eigenvalue problem. Then we found this one. Then using the singular value decomposition, we found AB. And there is a singular values there. And using this AB, we update environment tensor. Here is the left environment tensor. Of course, we have to update right environment tensor also. So solve this one and using singular value decomposition, find this one and update environment tensor, get the new eigenvalue problem and solve again. And then we keep this circle until everything converges. So Vector introduced this method in 2008. So this process is similar to constructing this bridge. When we try to make this bridge, we have to first construct the environment first, and then we keep this process, and finally we obtain the nice bridge. And it's comparable to Monte Carlo case. Monte Carlo case is they calculate here and there and over there, and they do simultaneously in many places, and then finally they connect. That's the Monte Carlo method. But here tensor network is quite different. Tensor network environment is very important in tensor network business. Okay, so this is the end of the introduction of numerical method. Let's apply the DMRG to one-dimensional Hubbard model. So one-dimensional Hubbard model is written in this form. So here is the hopping term and on-site interaction and chemical potential. Again, this is written as sum, and we can introduce matrix operator. Here, we have to use 6x6 matrix operator. This is this one. So there are infinitely many M is there and it's connected. So there must be some index contraction. Anyway, so there is M. But in this Hamiltonian, if I change the operator, C going to minus C, then Hamiltonian is invariant, nothing changed. So if here B is the same, B is the same side. So but B is this one. So B has an even number of Fermion operator, so nothing changed. But this one go to minus C and minus C delta and minus C this way. So there is some ambiguity. I mean, should I use M or should I use M star? M star is this one. So there is some ambiguity. How to resolve this ambiguity is easier. When the Hamiltonian acts on the basis, then we have to consider the sign. So this is the Hamiltonian, M naught product M1, M2, M3, and L minus 1. Here the site is total site, number of site is L. So this is H, and this is a typical basis. So when we calculate this one, this C have to go through this matrix operator product. So because of this commutation relation, when M and C data change, the M becomes M data, M star, and M star become M. So when this one goes through 0 goes to 0 and 1 go to nearby M1, then this the M1 must be M bar. So here the number of fermions of the left side is even, then this M bar becomes MM, and if it is odd, then it should be M star. So it's it goes through everything, then this M and M star appear. Okay. Then we have to consider Jordan-Wigner style consideration. I mean, look at this simple relation. The even odd, and if we use a 2x2 matrix, even odd, the diagonal is even and off diagonal is odd, then we make this contraction for multiplication. Then even is even and odd is even, and even odd is odd even is odd. So they keep this structure, even odd, and then keep this structure. And here even means just vacancy and two fermion. Odd means up fermion and down fermion. So the 6x6 matrix operator should be 12 by 12 matrix operator. Let's call it doubling. I mean, so the even basis acting on like this way, and odd acting on that way. And here is M, and there is M star. So there is even and odd. So this M star take care of this odd. So eventually, our matrix product operator has 4 times 12 times 12 times 4 tensors. So here's the physical state is 4, and bond the operator dimension is 12. Okay, so using this matrix product operator, we applied the DMRG, and we found the MPS. Then we can calculate the bond energy. So the bond energy is defined by that way. Just the summation I, and so let's consider only one bond. So there is a one bond, and there is infinitely long MPS. But using this property of canonical form is identical, so it can go up to there, and the left side go from here and there. So the bond energy is simply given by this diagram. So it's easy to calculate. So so I got the numerical result at mu equal 0 and u equal 4. this is the number, and the bond dimension is increased to 6 to 120, then the value is very close to exact value, and this exact value is known. I mean, the people solved this Hamiltonian completely. So this bond energy given by this one, and this value using this U equal 4, this exact value give this one, and I obtain this one. So almost 10 to the minus 5. Usually 10 to the minus 8 is they obtain this accuracy very easy. So 10 to the minus 5 or minus 6 is not bad. And let's skip. Let's apply the our method to another example. It's the Eisenberg. So again, this is the sum. I mean, the partition function is written in this way. You can see the sigma there, and this sigma is the whole spin configuration. So one, and this simple we use the simple relation. It's 2i2. It's e to the beta. Beta is the temperature, and this 2i2 is written as W times W, like this. So if we consider this quantity, then this partition function is written in this form. So clearly there is a summation, but here is the product. So diagrammatic notation is simply this is T and this is a connected OR. So it's just a single number. So single number. So as I said, the partition function represents the partition as a sum should be represented as a product there. So let's calculate this one. So how to calculate? As I said before, I introduced this MPS, and there is an MPO, and this relation, this is the eigenvalue problem. Then we can go, this state can go up. So using this relation, we can contract this partition function one by one, going up and up and up, up. So you can remove one by one if this is eigenstate. So let's calculate magnetization. So here is the T is the previously introduced the 4W product, and for magnetization we have to introduce sigma there. So there is a summation sigma. So T is the circle and S is the square. Then magnetization is denominator is a simple partition function, and numerator is almost partition function, but only there is a defect like S tensor there. Here is there. Then, as I said before, if we use the DMRG, I mean using the MPS, we can contract from bottom to top and top to bottom. So from bottom to top and top to bottom until we hit this defect like tensor. So we have to we get this this diagram. So Here, the left and right environment tensor is exactly same the environment tensor when we apply the DMRG. So, and the factor is cancelled out very easily when we go down, up, here, down, up. So the proportional factor, I mean the eigenvalue is cancelled out. So I calculate this magnetization, then I obtain this figure. So here line is the exact solution. Another result was obtained by C.N. Yang, 1952, quite long time ago. Anyway, it's the same thing. But you can notice there is some noisy, some instability. It's near the critical temperature. There is some noise because I use very short simulation time. If I use very long simulation time, then it may be smooth line is there, but there is some noise. But still, when we determine the critical temperature, this is a little bit difficult because this going down, 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 and 0 and 0, 0, 0. So it's hard to determine the critical temperature. So let's use another quantity. Another quantity is entanglement entropy. Entanglement entropy is given by this way. And when we apply the DMRG, we have to calculate the singular value. The singular value is lambda, and this formula will give us very nice pattern. So we can see very clear peak there, and so yeah, so it's easy to determine the critical temperature. So let's zoom in this peak. Then if and little bit decrease this bond dimension. So there is a when we increase the bond dimension, the peak is developed. So it clearly shows some critical behavior. And using this the peak point, we can make extrapolation. And we have finally we obtain this cutting point. We obtain 2.26922, and as I said, it's completely solvable. So we know the critical temperature. The critical temperature is this one, and this one is give us this way. So it's again -5 accuracy. We can obtain the critical temperature. Okay. This is the last slide in my talk. So let me summarize. I introduced 1D DMRG. So, we have to find the MPO. So given MPO, let's find MPX. So that's the general procedure of the DMRG. And we consider 1D Hubbard model. And let me emphasize again. We have to consider Jordan-Wigner style doubling. The sign must be taken care of. And we apply our method to classical 2D eigenmodel. And when we try to determine the critical temperature, the entanglement entropy is better than magnetization. And this one is better than that one. Okay. So, but so far it's more or less trivial. Actually, we have to develop the 2D DMRG. The 1D is almost well done, so it's almost trivial. So we have to, someone have to develop the DMRG, and if someone really make this method, then he can calculate the real genuine Hubbard model numerically. So I think solving this Hubbard model is equivalent to constructing this beautiful building in the world, I think. Yeah. So, yeah, thank you for your attention.
Thank you. Next question.
So, IC model, like this method can be employed for field, magnetic field.
Is this the idea for that?
Yes,
That's the idea for that.
with field, can you solve, can you do the same calculations with field?
I-
Magnetic field.
I think so. Yeah. It's not so. If I apply some magnetic field, then everybody has a little bit.
Yes, the matrix will still be 2 by 2 matrix with just a little modification, and but then,
Yeah, but
with two-dimensional IC model with field is not solved. So can you figure out from this what is delta, right? 1 by 15.
I think it's possible. So we can discuss later. And yeah, that's very interesting.
Yeah.
But I think if we construct this relation, then everything is the same.
Another question. So is there any specific restriction on this approach? I mean, for
Yeah, so the restriction is one dimensional.
Yeah.
Nothing but one dimensional, and it's a local interaction. The Hamiltonian should be local and one dimensional, so it's more or less trivial. Anyway.
but for the classical, you could do the for the classical case, you did the 2D.
Yeah, the classical is a 2D. So, I mean, the quantum case is 1D, classical case is 2D. And you know the equivalent. The classical of 2D is the quantum of this. So, but yeah, it's again, someone have to develop the 2D here.
Yes.
What is the difference between finite and infinite?
Okay. Finite DMLD is just the finite. The lattice size is finite. And infinite DMLD is it's the thermodynamic limit. The infinitely long lattice. And using this the invariant tensor from the infinite, then going up, that's the infinite DMLD. Finite DMLD is the zigzag that's going up and down. Swing left, right. That's the finite DMLD.
And which one is the better with the?
I think the infinite DMLD is better because I don't need the number of sites. Number of sites is already infinite. So the infinite DMLD, they use two scaling parts, the size and bond dimension. Here, only one bond dimension, time. We use the DMLD is a little bit better.
Okay, so if there are no more questions, let's thank the speaker. Thank you.
I will be talking about some checkerboard and zigzag patterns. These are patterns formed with coupled nonlinear oscillators. This is the work done with my PhD student, Manoj Varampe. See, these patterns are seen in numerous experiments. I mean, these coupled genetic oscillators. I could find several. Please don't ask me the details of these experiments. But I can tell you that there are numerous experiments because this is the obvious Turing instability is obvious instability in any reaction diffusion system, and they are seen in many contexts. Now, what happens here is that when you have up down pattern without any, I mean, this fellow is not x y x y. I mean, this is this may be x prime y prime, but this x is greater than y and another third side is bigger. In that case, what we do is that we assign some kind of spin. Spin in the sense that if second fellow is bigger than 1, then it has up spin. Second fellow has up spin. Third fellow is bigger than third fellow has up spin. Fourth fellow is smaller than third, and it has down spin. So if I looking at the local slope, positive or negative, I just say that these have these particular slopes. Okay. And if I don't have any reference to anything, so if I say this up down up pattern, then I will have this plus minus minus plus plus minus minus plus. And here there are two defects you can see. There are two places where there are two ups and then there are two downs. So there are deviations from the checkerboard pattern. I will say that these deviations have some kind of phase defects, and I will say that this is the order parameter. When this goes to 0, then there is a pure checkerboard pattern, and otherwise it is not. So, and if I look at pure zigzag pattern, and checkerboard pattern is simply you have zigzag pattern in both directions. So what you should have is the up down in first direction, the x direction, and up down in y direction. So I will look at the number of phase defects in row wise, number of phase defects column wise. I will add them, and if it is 0, then I will say that this is the checkerboard pattern. So, and this departure from this is the phase defect. So I will look at the sum of the consecutive spins. So if this particular, this plus 1 is a plus 2 divided by 2, etc., or minus 1 divided by 2. So this is, so number of defects. So number of any 2 consecutive plus 1s or 2 consecutive minus 1 is the number of defects. I will look at this number of defects in 1D and 2D, and I will say that this is my order parameter. Similarly, I will look at phase persistence. What I will see is that if the numbers site number 37 was bigger than site number 36, and during the evolution it remained so all along. If it was off spin, then it has persisted. So this I will call as phase persistence. I will use these definitions later. So this is an illustration of -1 + 1. So these 2 pluses is a defect. And if this +1 stayed persistence, I mean, okay, somebody who didn't marry even once or something like that, somebody who didn't change his religion even once, still this XYZ thing. So that is a persistent site. So number of persistent site, of course, this is a decreasing function, and how it decreases is a question we will address. Now I will be talking about this coupled map lattices. Coupled map lattices is a kind of, you can say, it's a cottage industry in India. I mean, non-linear dynamics persons, almost everyone has done at least one paper in coupled map lattices. So these are the maps. Some f of x is equal to minus. This is a Gaussian, shifted Gaussian, you can say, the Gaussian shifted by some particular constant. And this another logistic map. I'm sure that all of you have heard of it. So log mu x into 1 minus x. It shows period doubling, bifurcations, etc. So I couple such map. I mean 1 minus epsilon times. So it is the so there are sites 1, 2, 3, 4, and then each site 1 minus epsilon times some function. This logistic map of system. Epsilon by 2 times right hand side and epsilon by 2 times left hand side. I sum these and I update them synchronously. I update all of them simultaneously. Then you get new lattice values, and I continue doing this and this and this. So this is how I evolve. And then these are the 2 maps I consider. And I can do it in 2 dimensions also. So 1 minus epsilon times itself, and then up, down, right, left. I will take 4 contribution. I add and I update synchronously. So this definition of zigzag, and I mean this similar definition has been attempted with respect to time, but not space. And I will come to this. So if I look at coupled Gauss maps, and if I look at bifurcation diagram, what does bifurcation diagram tell you? The bifurcation diagram shows values of all sides. This is one dimension. In all values of all sides at some particular long time is given, and this does not from this diagram. You will not say that it will show some kind of zigzag pattern. Normally, up-down patterns, you will have 2 bands, 2 separate bands. There are 2 separate bands, but even when there are bands merged, you can see these zigzag patterns, and that is not obvious from this particular diagram. If you look at the pictures, then you will see this kind of pictures. So you will see that these kind of pictures are there. These are up-down kind of patterns have emerged. This up-down becomes down-up next time, and again become up-down, but still they remain zigzag, right?
So these kind of patterns emerge after a certain time. And I also look at the persistent side. So persistent side says up down becomes down up. I look at the persistence only at even times. So if I look at the number of persistent sites and number of this phase defects at as a function of time, then what I see is that over a long this thing, minus 2.61 to 1.26, minus 2.61 to minus 1.26. You can see minus 2.61. They're almost at the end of this big thing to minus 1.26. There is no way from the bifurcation diagram you cannot see that this will have a zigzag pattern. So what will be the special pattern? You cannot see when you look at all the values, right? I'm going in the wrong direction. And so you firstly you find these power law in defects, power law in order parameter in a range of parameters, something like self-organized criticality, not exactly, but not you don't tune to a particular value of parameter. There is a range of parameter over which you get this particular exponent. Okay, and at the same time, if you look at the persistence exponent, how many, suppose 3 was about 2 and it remained about 2, how many are such sites? Then it also shows power law, and this power law is 0.375, it is 3/8, which matches with Ising model, which matches with 1D Ising model at 0 temperature. So both these values match with Ising model at 0 temperature, and they are often over a range of parameters with this definition. So if I look at logistic maps in one dimension, you get the same exponent. This is a system which is studied from last 40 years. And then people have observed that there is a zigzag pattern, but you can, if you look at quantify it and how this zigzag pattern emerges, okay, you get this 3/8 as well as half, okay, over a range of parameters. So not just at a particular point, not just a critical point, but you get it over a range of parameters. So this is a surprising thing. You look at bifurcation diagram. The bifurcation diagram, there are 2 bands, but having 2 bands does not lead to zigzag pattern. Not having 2 bands does not mean that there will be a zigzag pattern. So I don't have time to give counter examples, but then there are asymptotic patterns. If you look at this, you can see this is a fellow which has defect and this is a fellow which does not have defect. So we are looking at these 2 cases here, distinct. And if you look at how the defects move, they are looking like annihilating around random walkers in time, and then they move around and annihilate on meeting in 2 dimensions. If you look at 2 dimensions, the order parameter decays like 0.45 and again over a range of parameters. And the persistence exponent is 0.22. Both exponents match with Ising model, and this is a big surprise. Why it is a big surprise? Because order parameter decay is like Ising model, okay, fine, over a range of parameters. Order parameter decay will be 0.45 at the critical temperature input. And persistence, you will see only at non-zero temperature, because only at 0 temperature. Because at any non-zero temperature, the spin will flip. If the spin flip, persistence will flip. So persistence is expected to decay exponentially at any non-zero temperature. And you know that the critical temperature for 2D Ising model is non-zero. So you cannot have a nice persistence exponent and nice order parameter exponent at the same temperature. Right? Because when you have a finite temperature, so this is a very interesting thing. And the lattice, if you look at, I have taken care of usual how the domains person. So you change the spins of one of the sublattices, and then this becomes bigger and bigger and bigger, and the entire lattice becomes this particular course. This is the way it changes. In logistic map, again you have in 2D, you have got the same exponents over a range. The range is from epsilon equal to 0.123 to epsilon equal to 1. So there is a large range over which you get order parameter, which is going like 0.45 and persistence exponent, and then again growing domains looks like this. Now, so there are 2 things which are surprising. One is that this particular exponent, I mean, it's not like, see, if it was 0 temperature, see, at any temperature below critical temperature, the correlation length is, I mean, you expect to not 2.16. Okay, it should go like half. It should go like half, but it just goes like critical temperature is like 0.45, and the persistence exponent is 0.22. The persistence exponent 0.22 matches with Ising model at 0 temperature.
the order parameter exponent of 0.45 matches with Ising model at critical temperature. So you have got both the properties in one go for this particular thing. So just to emphasize it again, at the critical point, the correlation length goes up, Z goes 1 over Z raised to half where Z5 is 2.16. Anywhere below critical temperature, it should go like 1/2. So we spent, I mean, I spent a very long time talking with the student that it has to be half. But somehow we are very sure that it's 0.45. So it is 0.45 over a large range of values. Okay, and time needed for complete thermalization is L raised to, so we also did finite size scaling, and it goes like the finite size exponent is 2.16 in our case, right? So there is excellent scaling collapse at 2.16 for both for order parameter as well as for persistence exponent, and the same dynamic exponent for persistence. So we just represent an interesting link between Ising model and nonlinear, non-equilibrium systems. Such as, firstly, it is unlike Ising model, it is synchronously updated. Ising model is 3/8 is random asynchronous update. So it is synchronously updated system, and maps don't have any Ising symmetry. There is no reason why there should be any link between Ising model and this thing. But there are same exponents. Firstly, they will get power loss over it, and the powers are similar to Ising model. And I think this is a very puzzling kind of thing. And this work is published in Chaos Solters and Fractals.
Thank you. The floor is now open for questions, comments. I have a question. So you showed this persistence exponent. So what is the equivalent quantity for Ising model? I mean,
Ising model that the box remains up during the evolution. And since at any finite temperature, it can automatically become down. The persistence is exponentially decreased at any finite temperature. The persistence exponents are only for 0 temperature, and it is going to be 0.22. And this is what we get.
So into that there is a older work by Satya and co-workers, I think Satya and co-workers. So into coupled plane lattices in 2 dimensions. If you replace something.
It has even older work, and he touches upon this particular older work by Hughes and Miller. And Hughes and Miller have a map which has the Ising symmetry, and it has this is the f of minus x is equal to minus f. That symmetry is there, and they couple it in 2 dimensions. And then first Hughes and Miller said that they have Ising type of thing, and then Satya said that it is not to do a synchronous update. But if you do a synchronous update, in that case, it is Ising line. That is what
I think so. Already there is a Ising university hiding somewhere. No.
There is an Ising university hiding somewhere in the sense that there is a negative slope around the fixed point. So this goes down, up goes down, and down goes up. Say that. And the fixed point has a negative slope. The map, the fixed point has a negative slope. So there is some kind of that seems very general.
so any other question? If not, let's hand the speaker again. Next talk is by Dipanjan. Dipanjan from the University of Warwick.
Hello, can you hear me? Good afternoon, everyone.
Mic is off.
Good afternoon, everyone. Chandran, I'm a postdoctoral fellow at the University of
Sorry,
Surrey.
your
Just check the
mic.
mic.
Mic is off.
You have the mic. There.
Okay.
Hello.
Yeah.
So I'd like to thank the organizers for giving the opportunity to present my work. So I'm going to talk about the nucleations in the presence of static and dynamic impurities in the gases. I'll give some brief overview of the model. So nucleation is very commonly observed in nature, and it has a lot of industrial application in the sense that can use it's a key step in the synthesis of new materials from the constituent of its particles. And simple lattice gas models can be used to study to get some insights about a complicated model showing nucleation. Here is one example where the authors have taken the IV lattice gas and they have studied the nucleation in the presence of pore, of rectangular pore, and they have varied the pore width and calculated the estimated the nucleation rate, and they have found this interesting result. They have found that for certain width of the pore, the nucleation rate is maximum. So this is an interesting result in the sense that this will be used to optimize the production or the synthesis of certain materials from the solution. And this model is the simple lattice model can also be used as a verification of classical nucleation theory and also study the average cluster states as a function of temperatures.
So can you define us what is the nucleation rate? How can we?
So nucleation rate is starting from white phase or the solution phase. That is of total time that takes to get a very big cluster larger than the critical cluster size. That's called the nucleation rate.
The density is conserved.
So it's a grand kind of nucleation. So yeah.
So you keep putting particles.
Yeah.
And critical cluster size is the gap between the two.
The critical cluster size is if you plot the droplet free energy, the maximum of the free energy is the critical cluster size. Like the barrier, the side actually the barrier. And but in this talk, I'm going to present the role of impurities in determining the nucleation rate and also the critical cluster size. So here is my model where I fully isolated gas in the presence of external field H, and also we have impurities in our system. So we have three types of particles: red, which is spin +1, which we call solute, and then white, which will spin -1, called solvent particles, and impurities are denoted by blue, which has spin +2. And these are the interaction. Epsilon plus is the interaction between impurity and solute. The epsilon minus is the interaction between impurity and solvent. And this is epsilon plus, this impurity and solute, and this is epsilon minus.
So in the Hamiltonian, SI is +1 -1, and in the Hamiltonian,
Yeah.
it's only +1 and -1.
Yeah, it could be anything. It could be +1, -1, or +2.
So for impurity, we'll experience a different thing.
No, for impurities, does not have any field actually. The field is explained by only by the plus and minus.
But if the summation over SI, no? So if SI is 2, even that term we got.
Yeah. So yeah, that's if you write proper Hamiltonian, it's very big Hamiltonian. So, but you have to exclude the impurity. And all for the remaining interaction, it is just the solute or is -J, solvent -J, solute is +J, and we set impurity interaction to be 0 throughout the model. And here is the algorithm. We use Metropolis spin tube algorithm at constant temperature to evolve plus and minus spins. And we also define some mobility parameter alpha, so which is used for the movement of the impurities, and it is a spin swap algorithm. That means we randomly choose an impurity, and then if the random number is and also all a random number uniformly distributed from uniform distribution, and if that random number is less than mobility parameter alpha, we do the Kawasaki or the spin swap dynamics to the nearest neighbors. If the random number is greater than alpha, then we use this random spin update algorithm, like the exchange plus into minus. So here I'll present some of the findings in my previous paper, where we consider the static impurities. That means impurities are immobile, they are fixed in the position, and their position is chosen randomly. And they interact neutrally with the both impurities interact neutrally with both plus and minus spin, that means solute and solvent. And here I have shown the droplet free energy as a function of cluster size. Lambda is the cluster size, and in the y-axis I have the drop free energy, and the rho I is the concentration of my impurities. So you can see with increasing impurity density, you see that the free energy barrier height as well as the critical cluster size both decreases monotonically. And why this decrement happens? To understand that, we treat that free energy obtained from the numerical simulation to the functional form given standard free energy functional form obtained from classical equation theory, and with all the parameters fixed, but we only vary the interfacial tension term. And if you do that, then you get nice fitting. So you can, the fits are so nice that you cannot distinguish, because here the fits are like this functional fit with varying A1 is also true, but you cannot distinguish. And here is the interfacial tension that we observe, that we obtain from this fitting as a function of the impurity concentration, and you see the monotonic decrement in the interfacial tension for the impurity concentration studied in this work. So that means only the decrement in the interfacial tension is solely responsible for the decrement in the barrier height and critical cluster size for static impurities. Now, natural question arises: what would happen if we consider some non-zero interaction with the impurities? So that means now we have non-zero interaction with impurities, and also we consider the static impurities, and the free energy. It can be shown that droplet free energy, the static impurity, depends only on sum of J plus J minus, and you can see that with decreasing the interaction stress, the free energy barrier height decreases. That means that impurities act as a, like, for positive Js, impurities act like a nucleating agent, and for the negative Js, impurities act as like they repel the solid forward particles, and for that we see the barrier height also increases. However, so in this case the concentration is rho I equal to 0.004, and you see for very large negative interaction there is a saturation in barrier height with respect to this interaction strength. However, in the case of when you increase the impurity concentration, we don't see any saturation in the barrier height. This happens because if your impurity concentration is high, then the gap between the two, the average separation between two impurities is low, and there is not enough room to fit a critical size without any overlapping with the impurities, and that's why you don't see any saturation in the free energy barrier. Now I'll show you two movies of the dynamic impurities for two different parameter values. So this is these two set of parameter values. We see that here impurities are preferring seat at the boundaries of the nucleus, and here they are doing some different behavior. They are itself doing clustering. So for the whole parameter range of epsilon plus and epsilon minus, something the different things are going on, which we distinguish in this behavior map. So this behavior map, I have along X, I have solid interaction, impurity solid interaction energy, along Y I have impurity solvent interaction energy, and I plot the fraction of impurity, average fraction of impurities that sit at the boundaries of the nucleus. And we see we roughly divide the whole behavior map in 4 regions: 1, 2, 3, and 4. 1 corresponds to the region where impurities prefer to sit at the boundaries, or the impurities act as a surfactant. The region 2, where impurities are completely excluded from the cluster, and because of the strong entangling interaction. And in region 3, which is scattered region, you see the impurities are itself forming clusters, and some of the clusters lie at the boundaries of the nucleus. And in region 4, the impurities act as a nuclear, and it promotes the nucleation. And in this case, nucleation rate is very high. So you see multiple clusters in region 4. So now what happens to the behavior map if we increase the temperature? We see that the density, like intensity of the region where it was acting as surfactant is decreases. Intensity decreases. It happens because of the high thermal energy. Now the impurities it gets homogeneously distributed in the system, and for very high temperature, you don't expect to be the surfactant effect. So now I'll show you what happens in the case of dynamic impurities when the system is antisymmetric. That means epsilon plus is exactly and opposite to the minus epsilon minus, and it is noted by minus 2j. And here you see that with decreasing the interaction strength or beta j, you see a decrement or increment in the barrier height. So here also for positive beta j, the impurities attract the solid particles, and for negative beta j, impurities repel the solid particles. And since impurities are mobile in this case, for very high negative beta j, impurities are completely expelled from the cluster, and it does not take part in the growth of the nucleus. And we see the saturation in the barrier height, which that means if you go to minus, like very strong negative, I mean minus negative beta j, you don't see any increment in the barrier height, which is not the case in the case of Statphys. So we also verified the saturation using some independent result, independent calculation, which is the calculation of the nucleation rate using forward flux sampling. Here also you can see the saturation in the nucleation rate. So the thing to note that since we see the nucleation rate as saturation as a barrier, as the nucleation rate is the key factor in determining the barrier height is the key factor in determining the nucleation rate. And as we see the saturation in barrier height, it is also reflected in a nucleation rate. Yeah, so here I plotted the nucleation rate as a function of interaction strength for both symmetric and anti-symmetric cut along the behavior map. So along for the symmetric case, I don't see much change in the nucleation rate, but in the case of anti-symmetric case, we see a huge drastic change in the nucleation rate, and it also saturated for very high negativity. And another thing is that we also observed some cross nucleation, like in region 4 of the behavior map, which is where the impurities itself do clustering, and because of this strong repelling interaction, if they share common boundary, that reduces the surface energy, and then because of that, the two clusters do the binding. Yeah. So that's the summary, and it's available in archive. And thanks. This is done with Professor Day3 at World. I would like to thank my funder and thank you.
Questions? So if there are no questions,
So the impurities are also changing, right? Means the number of impurities.
No, but it's constant. Impurity concentration is fixed.
But in your those the snapshots and also the movies, I can see this.
They are only the red particles, like the number of red particles is affected. That means we only simply is doing it. That means we can only see the red particles at the end, but not the impurity. They only do the movement.
Okay. So in your Metropolis updation, step, you only switch the +1 -1.
Yeah. And impurity, for the impurity movement, I randomly select an impurity particle, and which is less, random number is less than alpha for the mobility parameter, we go to the nearest neighbor, randomly choose one of the 4 nearest.
any other questions? Okay, so let's thank the speaker again. And let's thank all the speakers for this session. So, now there is a museum and archive visit, and we'll reach tomorrow in the morning.
Yes, so I think it
Yeah.